Good afternoon, and thank you very much. My presentation today is entitled Hormesis, Scientific Foundations, Mechanisms, and Biomedical Applications. Now, I'd like to first start off by defining what I mean by hormesis. As far as I'm concerned, hormesis is a dose-response phenomenon characterized by a low-dose stimulation and a high-dose inhibition. It's a non-monotonic biphasic dose response, and what makes it different perhaps than a general biphasic dose response is the specificity that I'll emphasize today dealing with dose response features. The term hormesis actually came into the peer-reviewed literature in 1943 by two University of Idaho mycologists who were studying extracts from the red cedar plant or tree on the growth of various types of fungal strains and observed a biphasic dose-response relationship. And so they adopted the name from the Greek meaning to excite. However, they were quite clear that there were many historically prior observations of this, and that the term itself had been known by various other terms such as the Arndt-Schultz law or Hoope's rule, other types of terms, but basically the term hormesis has become widespread within the literature. Now, in the world of toxicology, risk assessment, but basically biomedical research in general, there have been anyways what I'd call two dominant dose-response paradigms. In toxicology, the first one really was the linear, threshold dose response, in which you would have essentially no activity along a control, so compared to a control, background noise, and then you somehow exceed the threshold and you measure a biological effect. And that particular dose response dominated really throughout the early first half of the 20th century, perhaps the entire 20th century. However, but with the discovery by Dr. Hermann Muller of X-ray induced mutation back in 1927, and the follow-up research that suggested the occurrence of not a threshold, but a linear dose response. In 1956 rather, the U.S. National Academy of Sciences recommended for risk assessment purposes, at least with regards to radiation causing effects on the genome, that a linear dose response should be assumed. The very next year, a national committee within the United States, recommended the generalization of that concept of linearity to somatic cells, and that led to the application of linear models for cancer risk assessment. And so within the regulatory communities for assessing whether an agent is carcinogenic or not, there are really two different models that tend to operate. If it is a carcinogen, it's assumed to act in a linear fashion, and that has the implication that essentially there is no safe level of exposure. There can only be really acceptable risk. If it's a non-carcinogen, it's been assumed to act in a threshold sort of a fashion. And those have been the two dose response models that have dominated toxicology and risk assessment, and also probably various aspects of biomedical research as well. The Hormetic dose response is a third type of dose response. It doesn't have to be opposed to the other two. It's just a third type of dose response. And it's a biphasic dose response relationship. And we'll obviously have the focus on that today. This dose response is really the stylistic type of hormetic dose response, showing an inverted U shape of response. And we'll look at this more fully, but at the high dose, you have the typical decrement in function. But when you get to the threshold, which I'm calling a NOEL from risk assessment terms, which stands for no observed adverse effect level. But we could call that a threshold. You tend, in the Hormetic model, instead of having the response essentially regress and stay at the control response level, you have a flip to the other side and seeing a low dose stimulation. Ultimately, the stimulation will regress back to the control. And looking at it across dose, you see a low dose stimulation, high dose inhibition. The interesting thing that we'll focus in on, and that is that when you look at really thousands and thousands and thousands of these biphasic dose response relationships, the most significant characteristic to me is in fact that the stimulation is modest. When I first got into looking at this in a real detailed fashion, which was probably about, I'm going to say 25 years ago, and I tried to develop criteria for the assessment of this, I thought inherently maybe you would see these low dose stimulations that would typically go up to maybe 3-fold or 4-fold, maybe 5-fold more or so. Is this on?
Yeah, just have to get it closer to it.
Okay. I hope this is better. Okay, yeah, I think I can hear it. Okay, and I initially thought that you would see this maybe at the 3-fold, 4-fold, greater than background. But as I collected, I'd say hundreds to several thousand and several more than that examples of these biphasic Hormetic type dose responses, I didn't see that. I began to see that what actually characterized this response was in fact that it was modest, that it was essentially, yeah, sometimes you would see it with a stimulation exceeded twice the control, but that was kind of the exception rather than the rule. The rule was really in the 30 to 60% fold greater than the control for the maximum level, which really meant that it was pretty modest. It also meant that it was very difficult to study because many times it'd be very hard to differentiate it from background variability. And how could you be sure that it was real? And so you really had to get systems that had low background variability so that you wouldn't fall victim to saying you had a Hormetic stimulation when in fact it was just background normal variability. And so this was an issue that we'd have to come and take a look at. And so that's what this next slide is addressing. One thing that you find is that there is generally similar quantitative features with respect to the amplitude and the range of the stimulatory response. You can see this occurring, this low dose stimulation. It can occur if you follow it over time, either as a direct stimulation immediately after exposure, followed at higher doses by an inhibition, or in fact it could occur over time in which you have an initial inhibition or disruption in homeostasis followed by an overcompensation response. And so you knew just from looking at those two different aspects of it that there were two basic mechanism approaches that would be, in a qualitative sense, different. But regardless of whether this low dose stimulation was occurring with a direct stimulation or whether it was an overcompensation to a disruption in homeostasis or toxicity, the interesting thing to me was in fact that the quantitative features of the dose response were still the same. And that's going to become very significant in trying to understand this. And in fact, you know, if you have a low grade of toxicity, you really wouldn't expect to have a strong overcompensation such that you went 2 and 3 and 4 and 5-fold over that, because you're really trying to reestablish homeostasis. And so what you would, I think, want to have happen is that you would want to have homeostasis reestablished, but you'd want to make sure that it got back there. So you may have a slight overshoot. And that slight overshoot, then eventually you will re-equilibrate. And that slight overshoot actually is what we see here in the sense that the overcompensation stimulation is at maximum is really in the 30 to 60% zone. And so that's why I was wrong in my initial thinking that it should be many-fold over. That's really not what we see. The surprising thing to me was the direct stimulation is still within that same context. And that made sense to me later by saying that because I've seen now probably 40 or 50,000 different dose responses showing this with the same types of quantitative features. And I've come to a tentative conclusion that what the Hormetic dose response is actually showing is it's kind of a measure of biological plasticity and showing the capacity for gain within the system. And the capacity for gain within the system is actually fairly limited. It's limited to the percent gain. And that has very broad implications in terms of essentially anytime we're trying to increase biological performance. Because what that really means is that you're going to be limited with respect to the capacity to increase the parameter that you're trying to measure. Now, in terms of hormesis, what does that low dose mean? Well, it could be beneficial or it could be harmful, depending upon your use of it. And I believe that whether it is or it isn't should not be part of the definition of hormesis. Hormesis is really, I believe, should be seen just within a scientific framework. It needs to be, though, evaluated in a broader context, but that usually should come secondarily. Up here is an example of a hormetic type of dose response. I'll just say, well, you could use a chemotherapeutic agent like an anti-tumor drug. Now, at high doses, the anti-tumor drug should kill the tumor cell. What we've seen in really a number of studies looking at very large data sets, and that is that at low doses, low concentrations, a very large percentage of the anti-tumor agents actually stimulate the proliferation of the tumor cells. Now, the degree of stimulation is in conformity with the quantitative features of the Hormetic dose response that I just mentioned. If you take a look at the same kind of concept, but this time not with anti-tumor drugs, but with drugs that are designed to increase memory. Now, in this case, you're really trying to enhance memory. And what you find is that essentially all of these drugs, most of these drugs, induce it modestly, up to about the 30 to 60% zone at maximum. If you take, in fact, too large a dose, you'll find that the memory drugs become anti-memory drugs and will actually decrease the capacity of the system, the mice or rats that you're studying to actually learn the task that's at hand. How we study hormetic dose responses are really through dose response patterns, statistical significance, very significant with respect to extra emphasis on replication of findings, doing simulation studies before you get involved, especially if they're high throughput studies that you're evaluating, and mechanisms to try to deconstruct and reconstruct your dose response relationship. Now, as a general summary, before I get into show you some more specifics, basically I have probably 4 or 5 hormetic databases with many thousands of dose responses, indicate these biphasic dose responses using very rigorous a priori entry and evaluative criteria. And what I found is that the phenomenon is very general. It occurs independent of model, so you'll find it with plants, microbes, invertebrates, vertebrates, humans. You'll find it in vitro, in vivo. It's really independent of endpoint for the most part, although I would emphasize integrative endpoints. It's independent of agent and the level of biological organization. So you can see it at the cell, you can see it at the organ level, and you can see it at the level of individual. It's very generalizable within this context. Now, I've mentioned to you before that the low dose stimulation is modest. in this 30 to 60% zone. And I'll go back to the range in a second because that's going to be important. Basically, with regards to hormetic mechanisms, and in the, probably say 10 years ago, you could show people hormetic dose responses, then you could show them the replication of these and the generalization to perhaps more closely related biological systems. And even though you could have an observation that had high reproducibility, there was as you would know, there's a profound emphasis on proof that relates to trying to explain it mechanistically. And basically, what's happened in the last, I'd say, decade has been a major expansion in terms of explaining the occurrence of biphasic dose responses, hormetic dose responses, with regards to various types of mechanisms. And people have said, well, is there a single hormetic dose response? And you have to say, well, there really couldn't be, because if you see something that's independent of cell type, independent of biological model, independent of endpoint, it's really impossible to have the same type of mechanism. And so, but nonetheless, I'd say within the last year or so, I've put together about 400, maybe 500 different hormetic dose responses, which have been studied to the receptor level or cell signaling pathway level, in which you can, basically investigators have been able to identify the receptor that mediates the low dose stimulation, or identify the pathway that mediates the low dose stimulation. In a number of cases, people go much deeper than the receptor and pathway and come down to specific switching mechanisms that take you deeper. But at least at the level of receptor mediated and the level of cell signaling pathway, and using inhibitors to block the receptor and then removing them and seeing the stimulation occur again, or the same thing for inhibitors for cell signaling pathways, so that you can have reasonably clear evidence that the low dose stimulation actually is mediated that fashion. And I'll show you some examples as we get into this. Okay. Now, regardless of the mechanism, whether it's a receptor mediated, whether it's signaling pathway, whether it's non-receptor mediated, whether it's a direct or compensatory. As I mentioned before, the quantitative features of the dose response are the same. So it really doesn't make any difference what mechanism you have. And that's an interesting biological question, that the dose response features are the same. Now here's an example of the effect of platelet activating factor on the production of AMP in antigen-challenged guinea pigs. And why I'm showing this is that you have an inverted U-shaped dose response, which call it a hormetic dose response. And in this particular case, using essentially an antagonist for PAF, the antagonist actually blocks the low-dose stimulation, but it also blocks the high-dose inhibition. And so in this way, the same receptor is mediating both the low-dose stimulation and the high-dose inhibition. And that's an interesting phenomenon to me. The same thing is true here for the effects of corticosterone on prolactin production in these cells, these tumor cells. And here you have prolactin production being shown and the ERA receptor expression. And again, if you use essentially the antagonist for corticosterone, it blocks not only the low-dose stimulation, but the high-dose inhibition, indicating that this same receptor is mediating both processes. Over here you have an example in which that's not quite the case. We're taking a look at genistein on the incorporation of treated thymidine in these CACO cells. And the low-dose stimulation is blocked by essentially an estrogen antagonist that has no effect on the high dose inhibition. So this essentially you can separate the high dose, low dose stimulation, high dose inhibition. They're mediated by different means. Over here we have a histamine on progesterone production by MA10 cells. And here what we have is evidence that the low dose stimulation is mediated by essentially an H2 receptor subtype, whereas the high dose is actually mediated by a different receptor subtype. And this is the same kind of phenomenon taking place here, looking at the effects of noradrenaline on renin release. And here we have the low dose stimulation being mediated by beta adrenergic, whereas an alpha mediating the high. And so basically the hormetic dose responses, they also can depend upon the physiological state of the organism. For example, low doses of ionizing radiation, it's actually really interesting. Low doses of ionizing radiation, if you have inflamed tissue, can induce an anti-inflammatory phenotype. And this is used by especially German arthritic clinicians to treat arthritic conditions, that if you have an inflamed state, low doses of radiation can induce the anti-inflammatory phenotype. And if you follow this, and they have followed this in 5 or 6 different animal models showing a very nice biphasic dose responses, optimizing on the low dose zone. However, if you expose the biological system that is non-inflamed to the low dose of radiation, you'll actually get an amplification of the immune response. So it's very much dependent upon essentially the state of the state of the system. Now, okay. It's important when studying hormesis, when you take a look at responses, that I believe, even though it's broader than this, that you tend to see hormetic responses when there is an integrative response that's evaluated. For example, cell proliferation, fecundity, tissue repair, behavior, learning, disease or injury resistance, preconditioning, aging, longevity. And in actually going back and looking at, and this is where I see this 30 to 60%. And I wondered, well, what about cell signals that are related to hormetic dose responses? How large could the signals be? And I've looked at this in probably about 300 or 400 different types of dose responses. And the signals themselves are greater than the 30 to 60%, but maybe by double. Sometimes a lot greater than double, but generally not much more than double. So there's a difference between a signal, and there's a difference it's compared to an integrative response in terms of the quantitative features of the dose response. Here are some pictures of biphasic dose responses that I believe are examples of hormesis. Here's behavioral one, looking at the acute effects of ethanol on overall social stress, the low dose stimulation, high dose inhibition. Here you have looking at an anxiolytic drug using the open arm entry maze test, in which this particular case, essentially what you're trying to do is you're trying to get the rodent to do something it normally wouldn't do, and that's spend time in a lighted zone because it's more comfortable in a dark zone. So the drug at low doses gets the rodent to spend more time in the lighted zone. At higher doses, it actually makes it even more anxiety-ridden. But essentially, this is a basic test that's used in the pharmaceutical world, and most of the drugs that I have seen in the preclinical testing for anxiolytic drugs show this inverted U-shaped dose response. I call it a hormetic type of dose response. Here we have an example of morphine, and it's being evaluated in this case with regards to a type of seizure and a PTZ-induced seizure response. And if you can increase the difficulty of this drug, PTZ, to induce a seizure, then it becomes a candidate molecule for an anti-seizure drug. And in this case, the morphine increases the capacity, makes it more difficult by about 25%. Essentially, every anti-seizure drug that I have seen follows this inverted U-shaped dose response indicative of a hormetic response. Here we have methanol on fruit fly longevity, increasing the survival by about 20% in highly reproduced studies. Here we have sodium arsenate on PHA-treated bovine lymphocytes, looking at lymphocyte stimulation. Again, you have this very modest but a consistent low-dose stimulation, high-dose inhibition. Here we have an example of a prolongation in lifespan in DB mice by low-dose radiation, and we're having basically essentially an increase by about 30–40% between the control and the treated. And this next slide here comes from that study. And this is a control group at 90 weeks. Actually looks kind of like my 95-year-old dad did before he died. Just kind of made it a long time, but was really afraid and kind of losing it. And these are comparably aged, and where the skin was, fur was shiny, they looked full. It's actually pretty striking that in terms of a low-dose enhancement of biological performance consistent with a hormetic interpretation. DDT is banned as a carcinogen, but actually, and at high doses it is. Looking at at foci formation is a good predictor of it. Japanese investigators looking at much lower doses actually showed the DDT to reduce foci, liver foci formation, showing the hormetic biphasic dose response. The largest mammalian study ever to date, the so-called mega mouse study with 24,000 mice in it, looking at the 2-acetylmethylfluorine, essentially shows a biphasic dose response. This was repeated in 6 different rooms. All rooms showed the biphasic dose response. The hormetic dose response...
Professor Calabrese, the time is over. Please.
Okay, I'll just go right to the conclusion. Okay, so basically, thanks very much for listening to me. I think you got the basic story that I believe Hormesis is a general phenomenon, and hopefully it'll have application to the work that you are involved in. Thank you very much.